11h23, poro de perna. Tem que fazer uns negócios assim também, né? Que tem a batata, mas por enquanto, bora fazer os squats, velho, que é uma coisa que eu faço, né?
Although the fact that most current Yersinia species today are actually environmental seems to suggest that the great ancestor was also environmental. It can be easy to forget that not all bacteria choose a life of parasitism. Parasitism is attractive because by parasitizing other species, you get to inherit the biology they worked so hard to build. A stable temperature, abundant resources, and wait, yeah, no, bodies don't let you just get away with this for free. So pathogens, of course, have to evolve a set of tools that will not only allow them to obtain the resources that they need from their hosts, but to also fend off host defenses and competition from other microbes. It's no wonder that so many bacteria prefer to hang out in the soil or in open ocean water. Moving forward in time, we have the last common ancestor of all human pathogenic Yersinia species, the shared ancestor between Pestis, Pseudotuberculosis, and Enterocolitica. So what deadly tools did this common ancestor pick up for it to be the great-grandfather of the Black Death? Well, listen up. It picked up genes that allowed it to grow on vegetables. Wait, don't click off yet. I know that's kind of lame, sure, but did you know this ancestor also had genes that enabled it to survive the harsh environment of the mammalian gut? And that's kind of interesting, isn't it? The great-granddaddy of Pestis and all other human pathogenic Yersinia species started off as a gut pathogen, potentially spread from animal to animal through the consumption of contaminated food. This is a very different lifestyle than the one I outlined for Yersinia pestis, and makes Yersinia species look a lot more like another pathogen I talked about in my microbiome video, Helicobacter pylori. I find the context in which pestis evolved to be mega interesting because not only was it the Black Death, it was also the black sheep of the Yersinia family, bypassing the gut entirely. But again, just because you can survive lettuce
Doce,
doesn't
isso cansa
necessarily
demais,
give
mas
you a free pass
será
to
que
the
presta fazer um
host.
solento,
The
velho,
last
fudido?
common ancestors between all pathogenic
Tem que fazer uma de
Yersinias
perna só, só que eu não
took
sei
a
como é.
crazy piece of DNA via horizontal gene transfer in the form of the first virulence plasmid we'll be talking about today, which I'm just going to call P-YAB. While we don't have all the details of how Yersinia picked this plasmid up, we do know a lot about what this plasmid does. This 70 kilobase plasmid encodes quite a few genes that are essential for Yersinia infection. It carries the very useful YAD-A gene, which improves the bacteria to bind to cells, enabling infection. This synergizes well with the invasive gene, a chromosomal gene that enables Yersinias to manipulate the host membranes to allow Yersinias to invite themselves in. But it's also got the blueprints for one of the most impressive feats of bacterial engineering to have ever emerged out of the primordial soup. The P-YAP plasmid codes for the type 3 secretion system, a genetic program that builds the injectosome, a key weapon Yersinias use to evade destruction by the immune system's first line of defense. Let's just take a second to marvel at this feat of bacterial engineering. Except just exporting a PNG off of Protein Data Bank won't do. No, sorry. No. We've got to enhance, zoom, blenderize. Life was better before Blender. The injectosome is a massive, complex structure made from over 30 proteins that acts as a molecular syringe. This machine spans the double membranes of Yersinias, and when they make contact with their host cells, triggers the injection of Yersinia effector proteins, which is just a fancy name for proteins that do stuff in the host cell. The injectosome is the primary weapon Yersinia uses to fight the host's first line of defense, the phagocytes, which include neutrophils and macrophages. Taking a look at this impressive structure, are any of you biology students realizing something? Is there something somewhat familiar about how this protein looks? You might not recognize it immediately, but the type 3 secretion system shares a lot of similarity with bacterial flagella. Yes, the very same flagella that enables bacteria to swim in their environment.
Doce.
In fact, this secretion system is hypothesized to have diverged from bacterial flagella hundreds of
O
millions
flagelo
of
parece mais
years
impressivo, né?
ago. Once the injectosome connects
Esse molécula é
the
sinistra demais, velho. Cabrão.
hosts, we see the debut
Como é que as
of
proteínas se
SWAN.
dobram na
And
forma
by
dessa?
SWAN,
Carai.
I mean the YAP effectors, a band of highly trained specialists evolved for a purpose. To pull off the grandest heist of them all, disabling the phagocytes of the innate immune system. Starting off, we've got YAP-N, the gatekeeper protein. This cool-headed cucumber works in concert with other proteins to block the syringe from firing prematurely, ensuring efficient
Yesterday,
injection
the
if and only if contact is made with the host cell. Once contact with the host membrane is established, this triggers a series of signals that causes the formation of a pore complex that actually connects the cytoplasm of Yersinia to their hosts. This has to be done in a very precise way, as opening holes in a cell, as you can imagine, is pretty dangerous. There are antipathogen peptides that are all about destabilizing membranes, and if you destabilize membranes too much, you could cause cells to rip apart in a process called lysis. If Yersinia's goal is to avoid destruction by phagocytes, why bother with such a precise tool? Why not just use this needle and pop these suckers? Lysis of a cell causes that cell to spill out all of its guts into the surrounding extracellular environment. These contents are often pro-inflammatory and will alert the immune system. If Yersinia wants to survive in a host, it can't risk being detected by the host immune system at large, especially early on in infection. No, brute force will not yield a preferable result. Only a surgically precise strike can ensure Yersinia's safety. Once the carefully constructed pore complex is formed, the gate is then opened for the fearsome four, the quintessential quadruplets YAP-H, YAP-E, YAP-T, and YAP-O, who work together to disrupt the enzymes that are required for cytoskeletal stability and phagocytosis. Phagocytosis is a complex dance that certain immune cells like macrophages and neutrophils use to engulf and destroy bacteria. But mechanically, this process is governed by the coordinated assembly and disassembly of protein networks made of actin, whose structure and dynamics are reliant on certain enzymes. By disrupting the flow of actin, phagocytes become unable to move their membranes, and more importantly for Yersinia, unable to phagocytose. YAP-H even goes the extra mile, shutting down certain immune signaling pathways that stop the oxidative burst, a process that neutrophils and macrophages need to make the dangerous chemicals they use to destroy bacteria. If you want to learn more about neutrophils and the amazing oxidative burst, check out my spotlight on the neutrophils' tragic backstory. Link is in the on-screen annotation and in the video description. Yersinia's squad of proteins ultimately disarms phagocytes who encounter them and leaves them in a terrifying state of paralysis. While phagocytosis is a mighty antipathogenic defense, it is not the only weapon these white blood cells have. Even while completely paralyzed, these cells can still send out potent signals called cytokines that alert and empower other immune cells. Is what I would have said if it weren't for the effector protein YAP-J. YAP-J is an acetyltransferase, an enzyme that adds on acetyl groups to the amino acids serine, threonine, and lysine. Since biology is just weird shapes bumping into one another, adding a whole chemical group to the host enzymes is going to shut down a lot of biochemical pathways, including ones that enable a cell to make cytokines. As a last-ditch effort to call for backup, immune cells have one more ultimate ace: the inflammasome. If conventional ways of calling for backup are no longer an option, this desperate play involves using the potent signaling ability of the inflammasome to trigger the loudest programmed cell death there is: pyroptosis. This programmed form of cell death supercharges the cell's ability to pump out cytokines, perhaps even in the face of suppression. But if you've seen my other pathogen videos, you might expect Yersinias to have an answer for this, too. The mysterious YAP-M managed to evade understanding by scientists for a very long time. We knew it was important because mutating YAP-M would transform Yersinias in some contexts from pathogenic terrors to complete pushovers. But this protein, unlike the other YAPs, does not catalyze an enzymatic reaction, making it much harder to study. Several breakthroughs in Yersinia research reveal that while YAP-M itself does not have catalytic function, it can hijack certain host kinases to add a phosphate group to pyrin proteins. Pyrins are proteins that make up the
Fiz 11, fiz 12.
and by keeping
Quem
it
sabe?
phosphorylated, pyrins become unable to assemble. Paralyzed and with no way to call for help, what was supposed to be the first-line responders to infection instead become sitting ducks. As phagocytes lie there helpless, YAP-J triggers apoptosis, killing macrophages silently, a much more preferable outcome to the loud signals lysing cells produces. Apoptosis is a programmed form of cell death, one that can be healthy and normal under most circumstances. Apoptosis causes a cell to collapse into neat and separated membrane packets, trapping their inside guts and preventing unnecessary aggravation of the immune system. Yersinia takes advantage of this tidy cell death to stealthily move through the body uncontested. What I find so incredible about the YAP system is how carefully engineered this system appears to be. Through gradual refinement, each one of these proteins, for the most part, plays an essential role in Yersinia pathogenesis. Every protein has its function, and without this complex, coordinated heist, Yersinia would not be able to escape the wrath of the immune system. If you're enjoying learning about the complex molecular biology of the plague so far, consider joining membership. You get emojis to use during my live streams and access to non-science content. I believe all of my science content should be free for everybody, but your support really helps make that happen. Now, back to the plague. While the P-YAP plasmid makes pathogenic Yersinias a force to be reckoned with, not all pathogenic Yersinias are created equal. Enterocolitica branched off, doubling down on the last common ancestor's lifestyle, becoming very environmentally hardy and infecting the GI tract of an extremely wide variety of animal hosts. However, unlike its more dangerous cousins, Enterocolitica only produces extremely mild and self-limiting disease. An upset tummy that gets better on its own is a very different disease that essentially mauls you from the inside. And while pseudotuberculosis is nowhere near as deadly as its child pestis, it still causes severe illness in humans. The disease that this bacterium causes is called Far East Scarlet-like fever, a much rarer infection that shares pathological features with tuberculosis. Tuberculosis is known to cause granulomas, tissue structures that
Fez o som estranho no computador.
corpses
Sentiu?
of various immune cells, primarily the macrophage. But unlike
Como se
its
as pifar na
deadly
CPU de
disease,
novo. Caralho.
tuberculosis does not cause granulomas within the lung, but rather the spleen, the liver, and lymph nodes.
10%
Pseudotuberculosis
produzindo
is a
vídeo.
hardcore infection could be attributed in part due to doubling down on hand-to-hand combat with its
12
hunters.
minutos.
Enterocolitica is pretty happy with passing through the mammalian GI tract unbothered, and while relative pacifism worked out all right for Enterocolitica, pseudotuberculosis had carved out a path of violence, choosing instead to invade cells it didn't immediately destroy. Pseudotuberculosis developed several tools that more finely developed its hand-to-hand
Foi
combat
o SSD
capabilities.
sendo usado.
These tools include resistance to cationic antimicrobial peptides, compounds that all sorts of cells make to suppress
Tá
bacteria.
lagando.
Special adherence proteins that improve sticking to macrophages and inhibit phagocytosis, and proteins that enable the bacteria to resist the toxic insides of macrophages. This is just my speculation, but it appears the last common ancestor between pseudotuberculosis and Enterocolitica had a choice. Treat the mammalian gut as a nice
Essa
resort
season zero agora do
vacation, or
nada.
try to make it a more permanent home. This doubling down on violence can be exemplified by Asian strains of pseudotuberculosis making the superantigen YPM, a toxin that drives T cells insane, contributing heavily to the severity of symptoms for the host. Pseudotuberculosis provided Yersinia pestis with an excellent scaffold design to evolve from, a canvas that evolution would paint a sinister shade of black. It was already a brutal bacteria with the ability to turn the tables on its innate immune cell hunters. So how did this already formidable foe become the record-breaking killer we know today? It's kind of hard to know how to tell this part of the story. We don't exactly have a neat timeline of when Yersinia pestis picked up and lost certain genes. So bear in mind that I will be, to a certain extent, narrativizing these genomic changes. I wasn't able to access the source for this particular claim, but there is a hypothesis that changes in the natural environment may have caused changes in the population or behavior of certain rodents. This may have triggered an increase in the frequency of contact between parasitic fleas and rodents. Fleas could have picked up pseudotuberculosis from infected rodents, exposing the bacteria to a brand new host environment. This model isn't exactly straightforward, as pseudotuberculosis is, again, a GI pathogen. If a rodent's got pseudotuberculosis in its blood, it's probably an unusually severe case of infection. But let's just say that this is true. Over time, repeated and consistent exposure to this new environment would select for beneficial traits that would enable Yersinia's ancestors to be better suited for transmission in blood instead of through food. On a more macro level, Yersinia's ancestors would have to evolve specific accommodations for its flea host as well. These evolutionary pressures to escape the GI tract and thrive in blood, as well as the esoteric changes made to better suit the flea, would give rise to the deadly plague. Yersinia pestis, having been flung repeatedly into a hostile, unfamiliar host environment, picked up a gift in the form of the PMT1 plasmid from its friendly neighbors. Where or who the donor was is a little unclear. At least one group thinks that based on similarities between this plasmid to plasmids found in other bacteria, Salmonella was potentially the culprit. Salmonella may have donated this plasmid to pseudotuberculosis while they were both cohabitating within the rodent. Over time, pseudotuberculosis would make this plasmid its own, and this may have truly kicked off the speciation event that gave rise to Yersinia pestis, something that happened shockingly recently on the scale of biological history. This plasmid's got a couple of nifty tools that enables pestis to make a happy home in the flea gut. The first gene we'll talk about here is the YMT gene, which codes for a phospholipase D that neutralizes toxic metabolic byproducts that the flea makes. Like any good tenant, Yersinia pestis evolved to care for its home. Pestis had its own urease gene pseudogenized, meaning a mutation in the gene for urease made the protein non-functional. Urease is toxic to fleas, and so breaking the gene that makes urease prevented pestis from poisoning its host. Pseudotuberculosis and Entericolytica love making urease. It's crucial to maintain bacterial survival by balancing pH in the mammalian gut. But in the flea, all this does is ruin where they're squatting. It's not enough for pestis to simply tolerate the flea gut to become the plague bringer. Pestis has to complete the circuit and leave the flea to infect other animals. To do so, pestis must colonize the region of the flea closer to its mouth, the proventriculus region. Colonizing this area of the flea is not easy, and pestis can only do this through the production of a nasty biofilm. The regulator of capsule synthesis genes are genes that
lento,
repress
viado. Perca
biofilm
a contagem, isso foi
formation.
12.
Biofilms have come up time and
Fiquei
time
errando
again in
tempo
these
também.
packaging videos, and for good reason. They're nifty matrices of proteins and sugars that can shield its inhabitants from the immune system, as well as coordinate pathogenic behaviors across a population of bacteria. Bacteria generally need robust control systems to only begin biofilm production when the situation calls for it, as synthesis of biofilms is a pretty resource-intensive process. Pseudotuberculosis generally keeps its biofilm production under tight scrutiny. Pseudotuberculosis is more invested in invading cells and spreading by exiting the GI tract. It doesn't really need to invest resources into making a static extracellular structure like a biofilm. Why have biofilm genes at all then? Biofilms enable pseudotuberculosis to shrug off environmental threats, which explains why pseudotuberculosis only makes biofilms at room temperature, a signal that the bacteria is outside of its mammalian host. Yersinia pestis, however, lost a key regulatory element in RCSA to pseudogenization. A 30 base pair duplication within the gene caused an insertion of 10 amino acids within the protein, destroying its anti-biofilm function. Without this break, Yersinia pestis would be better adapted to making biofilms in the flea. Imagine the RCS genes as being a checkpoint tuned for the environment. Once certain conditions are fulfilled, the block on biofilm production is lifted and these pathogens can begin getting entrenched. In the flea, the requirements to lift this block are never lifted, making colonization of the flea near impossible. It turns out that damaging this checkpoint system by breaking RCSA is actually a good thing for Yersinia pestis, as it allows them to successfully colonize the flea proventriculus, perhaps at the detriment to its ability to survive outside a living host. Once pestis successfully colonizes the proventriculus, biofilms begin to build up, resulting in a condition known as flea blockage. And here's a warning. What I'm about to explain is gross. There won't be any real explicit imagery, but you can use your imagination. Flea blockage literally causes the gut of the flea to be blocked off from properly receiving a blood meal. Once the flea starts feeding, blood passes through the proventriculus, carrying it with it a mass of biofilm and bacteria, which reasonably causes the flea to vomit, bringing with it a mass of biofilm and bacteria. Of course, the flea, having just vomited, failed to acquire nutrition, and so it tries voraciously to feed again. Rinse and repeat until you have a mess of Yersinia pestis deposited directly onto the open wound of whatever animal is unfortunate enough to have been bitten. This is the dominant explanation as to how Yersinia pestis spreads via flea bite. But it wouldn't be my channel if I didn't acknowledge that the truth is rather complicated. Blockage of the flea is detrimental to the survival of the flea itself, as the flea starves to death, and it isn't super clear whether blockage is adaptive or harmful for pestis's propagation. It is undeniable that blockage is a part of the story, but let's not give Yersinia too much credit for being molecularly clever here. Pseudotuberculosis and Enterocolytica both were motile bacteria, meaning they can move around. Swimming is a pretty good feature for bacteria. You can move towards food and shelter. But what good is moving towards food and shelter when you're wrapped up all nice and cozy in a biofilm with food being hand-delivered to you by circulating blood? Pestis pseudogenized much of its flagellar machinery. It just doesn't need it anymore now that it moves with the circulation of blood. But this loss of flagella had a devastating secondary effect, removing one of the common targets the innate immune system uses to detect and defeat bacteria. This is just one out of an ever-growing list of anti-innate immune system tools Pestis picked up, and it was only allowed to give up its flagella once it stopped relying on living and spreading through the GI tract. The flea catalyzed a series of gene gains and losses that took the game plan pseudotuberculosis and turned it on its head, that transformed it from a bug confined to the GI tract to a bug that could infect and spread systemically, silently blowing up phagocytes along the way. But we're not even done there. Flea adaptations aren't the only thing that shaped Pestis's deadly nature. We now get to talk about the final virulence plasmid Yersinia picked up along its journey. The PPCP1 plasmid carries with it the PLA gene, coding for the protein plasminogen activator. This protein's role in pestis infection is actually not super straightforward. It can break down the host's fibrin, which are protein networks that help trap and limit movement of bacteria, enabling pestis to be freely carried through the blood unbothered. This has a side effect of slowing down neutrophils, as neutrophils rely heavily on fibrin networks to guide them towards infection. Under other circumstances, this protein is capable of catalyzing coagulation, which is kind of like the opposite of fibrinolysis, and I swear I do not have the synapse firing capacity to understand this right now. Subscribe if you want to hear more about all the other plague things I didn't get to in this video. So far, the genes that have been lost have all been pseudogenized, but this gene for acyltransferase LPXL was just lost entirely. This gene, along with the pseudogenized PAGP, helps the bacteria add the correct number of acyl groups to its membrane sugar LPS. In order to have a healthy and functioning membrane barrier, most bacteria need 6 acyl groups on these sugars. While this mutation would cripple most bacteria, in the very precise biological context Yersinia exists in, an acyltransferase mutation is well tolerated. This has an enormously lucky payout for Yersinia pestis. A tetraacylated LPS is highly unusual, and it loses its ability to bind TLR4, one of the premier pathogen-associated molecular pattern detectors the innate immune system relies on to combat infection. Not only is LPS broken in pestis, but its surface antigens are also protected from recognition by the F1 capsular proteins, which shield pestis from its immune cell hunters. This is the other gene on the PMT1 plasmid, and it codes for a fibrous shell that blocks phagocytosis, potentially by obscuring the surface area binding sites for its pathogen-associated molecular patterns, molecules the innate immune system can identify as harmful. While pestis is effortlessly solo-killing phagocytes left and right, it still has to contend with how deadly blood itself can be. Circulating within the blood are components of the complement pathway. This immunology nightmare pathway results in the supercharging of the innate immune system, and one of the final results of this pathway is the assembly of the membrane attack complex, which opens huge holes in the bacterial membrane. Pestis' ALE protein can jam the signaling required to initiate this program of cellular warfare. If you are keeping track of how much time is left in this video, you might be wondering why I haven't talked at all about the adaptive immune system, the more modern and more powerful defense wing. The thing is, Yersinia pestis evolved to so thoroughly destroy the innate immune system with stealth, precision, and overwhelming force that by the time the adaptive immune system is ready to fight, the battle is already lost. With innate immunity silenced and destroyed, the adaptive immune system loses precious time to activate. The plague kills in a matter of days, about the same time the adaptive immune system needs to prepare for battle. I made this argument in my video about the tragic backstory of the neutrophil. The innate immune system is actually really good at its job. A bacteria like pestis makes us all look like chumps. But know that for the most part, if you remove even one of pestis' virulence genes, the immune system cleans it up without a problem. Pestis exploded into history pretty recently, and mammalian biology just hasn't had the time to catch up. Thankfully, we don't have to. Modern pharmacology in the form of antibiotics like streptomycin and gentamicin, which shut down bacterial protein synthesis, can bring down the case fatality rate from a devastating 90 to 100%, all the way down.
Como é que vai falar que isso é um design inteligente?
Wait a second,
Quem foi que
that's
fez
still
isso
terrible.
conseguir se matar?
While horrifying diseases like rabies and excellent prognoses provided treatment has been initiated, current antibiotic treatment can only do so much for
Resultado da
cases of pestis.
competição livre.
Science is a bit behind the rest of the disease-fighting team. But this is where budding researchers like many in my audience can step up to bat. There are still many unanswered questions on how pestis functions in both mammals and in their flea hosts. Perhaps by uncovering more about how this bacteria functions, we can open up the surface area of attack for drug designers and vaccine manufacturers to put away the Reaper for good. Special shout out to Gecko and Rainapper for helping me with the Blender song. Shout out to Clockwork and Emil UX Lizard for helping me with learning Blender. And of course, thank you again to all my members for supporting the production of this video. Thank you all for being patient. You're all rock stars.
Nature is beyond amazing, and all around us. We got deserts, forests, rivers, mountains, oceans, the list is near endless. And while every habitat is breathtaking in its own way, there are those particular places that seem time and time again to captivate people more than normal. And there's perhaps no better example of this than the Amazon rainforest and river, sometimes collectively known as the Amazon Basin. This area is made up of extensive lush rainforests, swamps, and thousands of waterways, providing an abundance of resources, which allows for the existence of some of Earth's most unbelievable life. And its crazy nature has led to it often being touted as the last frontier on land. And this is further reflected by the fact that a poll with over 600 million votes listed it as one of the 7 new wonders of Earth. Impressive, and proof that nearly everyone knows about it. But that said, not all, or dare I say most, realize just how giant it truly is. The rainforest, for starters, is by far the largest on the planet, outcompeting the second place, Congo, by a magnitude of 3, while the Amazon River is so expansive and powerful that it moves water about 6 times faster than what's seen in any other river on Earth. In fact, you could combine the Nile, Mississippi, Congo, Ganges, Thames, and Yangtze River and still not beat the amount of water the Amazon is moving. And due to its sheer size, the Amazon
Foi isso aí. Esse vídeo de perna é uma bosta, velho. Tem que aprender a fazer direito esse negócio, mas é isso aí. Vai indo.